Okay, we're going to get started here. So welcome everybody to the final seminar on the Go with the Flow series. I want to thank Ramsey for hosting and running the last 4 seminars. I'm delighted to have our 4 speakers back. So Rashmi Bengali from KBI, Jay West from Bristol, Andres Martinez from Roche, and Raf Dadir from Janssen. And what we're going to do today is really have a roundtable discussion. I think we've covered a lot. I was really impressed with the breadth of the topics that were covered over the 4 seminars, from dynamic binding capacity work that Rashmi spoke about, to UFDF that Jay talked about, to the use of flow with malts that Andres went through, and then Raf finished up last week with the talk on fill finish, and actually looking also at filter performance and looking at the binding of proteins to filters. So I think for me, it shows the breadth and depth of process analytics technology. Obviously, we're talking about flow, but I think it's a bigger market that we're all working in and dealing with. And I think this industry, especially PAT, has come a long way in the last 20 plus years. I think the next 4 to 5 years, we're going to see a lot more progress. And I think the 4 individuals that you hear from, I would highly encourage everybody who's on, if you haven't listened to the seminars, you should take a little bit of time. You're definitely going to learn something, and you're going to see that these individuals are at the forefront of what's going on in PAT. So maybe with that, we'll kick it off, and maybe I'll start with Raf and Jay, and just maybe talk a little bit about the challenges you were facing and what you were trying to solve when you looked to evaluate the flow VPE technologies. Maybe start with Raf.
Yeah, thanks, Tony, for that question. I think when we were looking at flow VPE, right, so the challenges that we were really trying to solve was just the extent of how well do we understand the process that we have day to day in our manufacturing facility. So traditionally, we have a lot of offline sampling. We take a sample from this part of the process, we take a sample from that part of the process. So we're just capturing maybe, let's say, a very small percentage of what is really going on, and then having the ability to have online real-time monitoring. That's really the challenge that we wanted to solve. For us specifically, where we looked at flow VPE was that we were running a clinical facility, and very often it's the first time that product actually comes to a GMP facility or to that specific scale of manufacturing. And what you're looking for is really, besides manufacturing your product, you're looking really to understand how the product is behaving through the different process unit operations, and just collect as much understanding on the process and the product as much as possible, so you can optimize your process and get ready for commercial manufacturing. And that was really the challenge that we were trying to solve. How do we get in the best way all of the data we're looking for? And for us, I mean, the answer was really to go into PAT, and then specifically for protein concentration, we ended up going for flow VPE.
Great. And Jay?
Yeah, thanks, Tony. So I'll just be brief because I know we have a lot of questions to talk about, and people can ask if they want me to follow up. But simply, basically, it was the high concentration UFDF processes that we started working on in development about 4 years ago. It's 2017 at Bristol Myers. And they always require the concentration range during the process is very wide. It's between about 10 and up to over 200 grams per liter. So there's not really any other instruments available that can measure concentration in real time. I mean, you're measuring right now, and probably most people in the industry do it by mass, which is, of course, then you get large errors if you're using mass potentially, and extrapolating based on the starting mass and concentration to the ending mass and what the concentration would be based on the mass. It's much better to measure the concentration directly. And the flow VPE was really the only instrument I think available that could measure that large dynamic range at the time. And I think Belief still is relatively simple to set up too, like under other, like I know Raman and say FTIR can do that, but they require modeling. So this is a pretty fast system to set up. Thanks.
Great. And maybe to Rashmi and Andres, what What are your thoughts about using technologies like flow for different therapeutic modalities? So, gene therapy, or even, what we're seeing today in mRNA. Maybe start with Rashmi.
Yeah, so I feel, I see no reason why we cannot apply it to different molecules. Like we have used it successfully for different protein molecules. We have used it for MABs, bispecifics, fusion proteins. So I don't see any reason why we cannot apply these to other technologies as well, because I feel, like even though the molecules are different, the basic unit operations which we are using for purification. So since I'm a downstream person, I'm talking about purification. So we are still using chromatographies, we are still using UFDF. So everywhere where we are actually going to use the UV spectrophotometer, that can be easily, we can apply this definitely there. And, like talking about, viral vectors for gene therapy and, similar stuff. Okay, we still use affinity chromatography for the capture and all. We probably cannot use that for quantifying the vectors, but we can definitely try to have an application for the impurity measurement, concentration measurement. So I definitely see that we can extend the applications of this beyond what we are currently doing.
Okay, great. Thanks, Rashmi. And Andres?
Yes, I think as Rashmi said, there is a lot of potential with the technology. So not to repeat what she already said, I think another advantage is the fact that it's quite applicable to connect with other sensors as well. And I think that can be quite interesting, especially with AAVs, which can be challenging to detect. Maybe you can connect it with other sensors as well. Maybe you can couple the signal from your flow VPE also with the Raman or with other spectroscopic methods. So I think there is a lot of promise there.
Maybe, Andre, staying on that topic, what do you think has been the biggest barrier to adoption of PAT technologies, whether it's flow or it's Raman? What's been the pace has been relatively slow. It's definitely picked up in the last few years, but what's your sense of why it's taken so long for companies to start to implement technology like this?
But I don't know if I'm the most adequate person for that question. Maybe someone in manufacturing can better answer that. But at least the impression that I get is that, I mean, if it's worked so far, then like if you look at it from a project point of view, if you have to get a new technology into your project development and you have your timelines and you have your risks, then you are taking more risk than you want to, than you have to. And if it's worked so far, then why do you have to do it? So maybe the question is about the motivation. Like if you look at it from the personal project management point of view.
Yeah, no, that makes sense.
Maybe I can add to that, Tony. So from a pure manufacturing perspective, I think especially in a drug product fill finish, right? So people are always looking at removing things from the process to maintain sterility. So if you're going to start introducing additional pieces, that's something that, I mean, puts the the thrill in somebody from a QA group, right? So I think that's for sure a challenge that we are generally facing with. And I think on top of that, also just we are with PAT, we're in between, let's say, two traditional functions, right? You have the QC group that has really all of the know-how and the expertise on analytical testing, and then you have the operational group that has all of the know-how and expertise on the manufacturing shop floor, and you're bringing those two worlds together. And so there is a lot of ambiguity as well on who's going, I mean, how do you combine that skill set into a single person or into a single group? So I think that's also a challenge that companies are now starting to tackle, as well as to how can we train operators to have additional skills that they didn't have before, or how can we give QC staff, let's say, more operational expertise. And I think that probably adds as well to as to why we're seeing that there might be a slow implementation, let's say, of these new technologies.
And I assume it helps if you have dedicated groups within your company that are focused on I'll steal Jay's terminology from his seminar, the advanced PAT. Having that group must really help drive the evaluations of new technology. Maybe Jay, do you want to speak to a minute about how that group has kind of evolved and why you think it's been helpful in terms of driving sort of the evaluation of new technology and then the implementation of those technologies?
Yeah, so I guess you call it advanced, or I call it enhanced, maybe, which is kind of the same thing, in a sense. And I think it speaks to that these are they're pretty complex molecules, biologics, and especially if you are measuring CQAs, like especially compared to a small molecule, which is the API of a small molecule is the molecular weight's under, say, 500. It's almost completely pure substance. Sometimes it's just stereoisomer. But otherwise, a biological molecule, in contrast to monoclonal antibodies, extremely heterogeneous. There's hundreds of thousands of chemical bonds and atoms. So that's requires much more sophisticated analytical instrumentation in order to measure the CQAs. So protein concentration is a great place to start right here with the flow VPX. So what we're trying to do in our enhanced or advanced PAT group is take some of these tools that were first developed for, say, small molecules, or things that are, at least my focus within the group has been to try to utilize technologies that we could theoretically use in a GMP facility in the relatively short term, like say 5-year time span, because technologies that are not mature, I think, you're not going to take something that's completely a brand new technology and try to implement it in PAT at this point. We're going to try to take the most rugged and proven technologies, such as this VPE, which has been around for over a decade, or approximately, if you include the solo VPE, and put that into the GMP world. So that's sort of the enhanced PAT that we've been working on, is using proven, reliable, robust, accurate, precise technologies for our enhanced PAT, and then sort of go from the most simple to more complicated, maybe like mass spectrometry we talked about, Tony, in the introduction later. That can come probably at the very end, I think, because that's probably the most complex of all analytical tools.
Yeah, staying with you, Jay, on this one, but when you think about the way the industry has evolved, right, batch-based processing has been kind of the dominant kind of approach to manufacturing biological drugs. Continuous has definitely taken off. Obviously, upstream continuous has been around for a number of years. Downstream continuous is definitely, we're seeing a lot more folks in process development and moving into actually clinical manufacturing using downstream continuous. So maybe could you maybe speak for a minute on how you think flow VPE or flow VPX technology could be applied in continuous manufacturing, and maybe even expand it to talk about other PAT technology you think that could be very relevant to continuous manufacturing.
Sure, thanks for the question. So first of all, for batch, there's definitely an advantage to this technology because it can reduce the lead time for each one of your batches, as Rob sort of talked about in his last talk too, that you can, at some point we can have an automated dilution process control based on real-time measurements, where the process where you are controlling the concentration during, say, UFDF and then subsequent dilution to the drug substance. Right now, we are at the stage where we want to do process monitoring with the VPX, but for the batch processing, this is going to replace the offline measurements by doing inline measurements, so that's slowly going to reduce lead time. And once we get to the point where we can measure real-time inline and do real-time in-process release, then I think true continuous could actually be something that's realistic and possible, at least for the UFDF part. And I think this true continuous is going to be, I think, difficult, linking in from upstream all the way through downstream. But the way our process development group has gone about the problem here is you use elements of continuous. So they were looking at a continuous protein A capture, continuous polishing steps. Now I think that we're on the cusp that we're maybe in 5 years we could look at a continuous UFDF process. And I don't think it would be possible without inline A280 monitoring.
Yeah, and I think that, you know, the whole continuous from upstream to downstream, the first step is definitely going to be the continuous capture step on protein A. Maybe Rashmi, given all your experience in that area, what's your sense on using FlowVPX or other PAT technologies in that continuous capture where you're using multi-column chromatography for capture or multi-cycle?
Yeah, so that's, I think, a very good question. So, you know, like over the years, like there has been so much of improvement in the upstream processes, and, you know, continuously, you know, the input to the downstream processes sort of changing. And then when we are talking about, you know, operating protein A on a large scale in continuous cycles, I think it's very necessary that we have to understand that some type of changes can take place in the process. So there is a variability which can occur maybe in the upstream process, or there may be errors in the titer determination. And so we cannot stick to a fixed value, like of binding capacity, for example. So we are continuously loading to a fixed value. But if we can implement this flow VPE downstream of our columns, we can actually control, you know, like the start and stop of our loading based on what we are getting. And this can also provide us a sort of warning and indication that, okay, our resin is degrading, okay, so it has, you know, completed its lifetime, you know, things like that. So it can definitely be applied, you know, for enhancing the capture process, given, you know, just to, you know, mitigate the variability which can occur upstream of the actual capture step. And for continuous chromatography, yeah, I definitely see that that happening, since, you know, as the capacity decreases with time, we can capture it real time during the manufacturing process.
Yeah, and maybe continuing on that theme, if you think about you know, the migration that's happening towards continuous, and Andres, maybe for you, your talk was particularly enlightening in terms of using multiple PAT technologies, you know, together to measure not only the percent aggregation but also the concentration of aggregates. When you think about technology, you know, where do you see the combinations happening? I mean, clearly you showed the combination of mols and flow VPE, but what other technologies do you think could be combined with flow that would give you more insightful information about your process?
So I think something that could benefit in general from the concentration measurement would be maybe, in some cases, for example, Raman, that you have an increase in background fluorescence because of the increased protein concentration. Then maybe there's a possibility there to scale down the signal that you get by taking into account the flow VPE information. I think that would be the application that comes foremost into mind. Maybe you could also use your concentration measurement if you have a varying sample going into an HPLC, so that you can determine how much volume you want to inject, or how do you want to treat your sample before doing an HPLC on that.
Great. And Ralph, how about from your perspective? I mean, clearly you guys are doing an awful lot of work in PAT. Have you started to combine technologies, or is it being mainly, you know, one technology evaluated at a time, or are you integrating technologies?
I think it's a mixture of both. So on the one hand, for instance, we're looking for applications where we take an existing tool and we're trying to see what that tool, in addition to what it has been made for, can be used. I mean, for instance, flow VPE is predominantly or almost exclusively used at 280 nanometer, but comes with the option to go from, let's say, 190 up to 1100 nanometers. So you have access to that whole UV-Vis range that contains a lot of additional information. So that's for sure an area where we are looking at as well. And then in addition to that, it's indeed the combination of multiple PAT tools where, let's say, the standard multivariate analysis, where PAT tool number 1 combined with PAT tool number 2, individually they might not give you something that you're looking for, but the combination, if both of them are trending in a certain direction, they could give you additional feedback on what is happening into the process. So for sure, we're looking at both of those two routes.
And maybe Raf, sticking with you on this one, when you think about the flow project, which obviously you were working on that for a number of years, how was the project justified, and was it based on was it dollar based, was it risk based? How was the sort of decision made on justification of the technology?
Yeah, and that's I'm smiling because it's a typical question you get always, right? So what's the business case? And I just maybe a side note, but I always think that if we keep on, like from an innovation and technology perspective, if we keep on using the same processes that we use for a business, then we will never kind of invest. We will always keep on investing in safe bets, right? So we will never kind of go off the beaten path. So, but it is a reality, right? That the business case question always comes back, and I think it's a mix, right? On the one hand, you do have tangible benefit. You can say, okay, I calculate a return on investment for flow VPE. So if I'm going to implement flow VPE, which costs that amount US dollars, if I don't pull the sample anymore and I have, let's say I get a certain lead time reduction, or I just have no person in the QC lab coming in at night anymore. So that's obviously the benefit I get, and you can really quantify a certain business value out of that, and typically that comes out positive. So that's at least enough to convince the base level of people that would need to sign off on that. But I think what we have been really trying to do is convince people of the added more, let's say, less tangible benefits. So if we have this additional data that could be used, for instance, to better understand the process, and that's going to lead to, let's say, a better manufacturing process at launch of that product. So it's very difficult to quantify that value, but it is something that we try to emphasize a lot, right? So can we, for instance, reduce our tech transfer timelines or scale up efforts that we need to do based on the tool. So if we have a tool, we're collecting data, and we can remove a lot of the offline sampling during a PPQ approach. So it's a mixture of both. But I do agree that there is still the reality of the business that you need to provide that business case value.
But it sounds to me, and maybe just opening it up to the whole group here, but it just feels a little bit more like it's a data-driven decision. In other words, you have an idea about where the technology can be applied. You go ahead and you run some experiments. You show the data, and then there's a discussion about, okay, well, we could implement this in a UFDF step, or we can implement this in a more universal, DBC for the protein A columns. Is that generally the approach that most people are taking, or the teams that are is that what they're taking? Is doing the experiments, seeing exactly what the data is showing, and then driving it towards a business decision?
Yeah, I mean, I can speak maybe from my experiences that we've had, let's say, companies where, I mean, we've had Ramsey come in a couple of years ago with the flow VPE to collect data, and that data was crucial in deciding on whether to go with flow VPE. Similarly, we've had companies that were unable to provide, let's say, a demo instrument for us to test, and then that led to us not purchasing the technology because we don't really want to purchase something of which we have no data. So, I mean, that data piece is absolutely crucial in providing or justifying that value that it brings to the business. Absolutely.
It's a shame Ramsey's on the call. He'll be using this to say, look how valuable and important I am in everything that I do. So just not a set of questions. So hey, Ramsey, actually over to you. Any questions from the audience?
Not yet, but I would just add, if anyone has any questions on the line, please type them in, and I'll relay the message, and we can have a discussion about whatever topics you'd like to hear from our panel. Thanks.
Yeah, so maybe moving to kind of more of a future discussion around PAT, and I think Raf, when last week when you were presenting, you talked about moving from monitoring to control, and maybe could you spend a few minutes just talking through what the challenges are going to be for even Janssen or other companies as you think about, it's one thing to be able to monitor. It's much more challenging to be able to say, put this into a control environment, and you don't rely on the human factor of saying, here's the concentration, you plug it into the formula in the batch records, and now you know what the exact number is, versus it's fed back in and the operator is told this is exactly what your next steps are. So maybe you could spend a few minutes on how you see that evolving.
Yeah, I mean, it's absolutely true, right? People get much more skeptical when you say, hey, I have this tool, I just want to monitor what happens in the process, and then I'll leave you to it and I don't bother you anymore, versus, hey, I'm going to use this data to actually drive GMP decisions. So there's really, it's the same tool, it's the same GMP process you're implementing it in to, but it is, the applications are completely different, and that comes really with a bunch of additional requirements, right? So you need to really, from a purely analytical perspective, you need to be 100% sure that the data that you're capturing is correct. So if you would be monitoring simply, and an operator would have, let's say, a wrongful assembly of the sensor, or maybe a calibration that's out of date, that's okay, because you might just be interested in looking at a certain trend, or just looking at the data, and at the end you might decide on how useful the data could be for what you're looking at. But if you're really using that into a GMP process, a lot of those actions become very important, right? So the analytical method needs to be spot on. It needs to be fully validated. Your data integration, your signal coming from the flow VPE, that has to go into a data historian for verification. So that connection needs to be spotless as well. And then you have the whole quality piece, right? So and the whole, let's say, roles and responsibilities around that, to make sure that your instrument is maintained to the right level. You have regular checkups by the supplier, or you have regular calibrations taking place, and all of that documentation. That's a lot of things that need to come into place from, let's say, within the company that's implementing the tool. And then you have on top of that just the external acceptance, right? So you're using this tool now to make GMP decisions, so you need to not only convince your own operators that this is something that is reliable, but you need to convince health authorities as well. So and in order to do that, you need to really have a good data package showing them that this is equivalent to or better than the current control strategy that you're applying for your process. So that could be either a comparability study or comparability protocol that you have outlined, but there is indeed a huge amount of data that needs to be collected to really fully convince the health authorities that this is, let's say, as that equivalent or better to the current way of working.
And Jay,
Hey, I had a
was
question from the line, if I can break in for a second. I have a couple, actually, and maybe this is open to everyone. What is the focus of your PAT strategy? Is it to replace IPC, endpoint release testing, add knowledge about your process, or something else? I think we kind of touched on that, but I guess maybe as part of the justification, when you first bring in the technology to get that data, what was the original point, or the main focus? I guess we could start with Rashmi there.
So, okay. So, as I mentioned before, the same thing, the dynamic binding capacity, the binding capacity of the resin, it's important. And if it's changing over time, application of PAT is definitely going to be, useful there. So, just to make sure that we are not underloading or overloading our resin, we are getting consistent, performance. So, maybe we have an output of the flow VPE or any of the PAT instruments attached to some algorithm, right, which is doing the control for you.
Right. So in your case, the main strategy here was to get better process understanding.
Yes, I mean, it is also to handle the variability which can inherently happen in any of the process, so that we have a control strategy that irrespective of whatever variability, we are still going to have consistent performance.
Cool.
Yeah, if I can just for us specifically, it also was that our initial challenge that we wanted to solve was that increased process understanding, right? But then from the moment that you have that tool, you're seeing a lot of data that you never saw before, and then it goes up to the point that you can start using it for other applications. Where now, let's say we're using that for an IPC, and in the future, let's say I talked about that last week for control of actively controlling the process, and then who knows in the future as well, potentially for release, right? So it is something that is not static. So you might implement FlowVPE for one particular purpose, and then you potentially see all of the applications that you could use it for in the future as well, and then kind of takes from there.
There's another question here. I think maybe Jay, this would be a good one for you. In the GMP environment, who owns the data and the equipment? The analysis QC or the process? So I guess the question is really who's, and I mean, it maybe makes sense to give the BMS perspective because it's kind of a big company. Who owns the equipment, or who's the owner of the analysis, I guess, in the case of inline system? I'm not getting sound from you, Jay. I'm sorry. So maybe while Jay's working on that,
Do you want me to quickly? Yeah, I can chime in. Then I mean, I can give the Janssen perspective on that. So it is indeed kind of, as I mentioned before, in between two worlds, right? So, but I think ultimately the process and the whole equipment should be, since it's embedded into manufacturing, the owner of the equipment, and then all of the qualification exercise or let's say actions that revolve around that should be owned by the people that normally own as well the let's say the equipment, the other equipment on the manufacturing side, whether that would be, let's say, the engineering group as well. If it's about data, then we just look at what's the application, right? So if the data is used for IPC purposes, in our case, that data is owned by the QC group. So that also means that the data coming from that PAT tool is owned by the QC group, which will then also do or will be responsible for the review and then the actual sign off on that data. If it's only for monitoring purposes, then it's generally the development group that would own that data. But for IPC, it would be QC, and that's kind of how we have settled that kind of a duality between it's an analytical tool but in operational shop floor. But so let's say from a hardware perspective, it's really owned by operations, but then the data coming off of it would be owned by QC.
Can you hear me now?
Yeah, Jay. Yeah,
okay.
Maybe you can add your perspective there.
Yeah, we haven't gotten as far as what RAF is in terms of in-process control. So I can't give you, but I know the equipment is owned by manufacturing, and in-process testing is typically owned by manufacturing. But for in-process controls, that's something that I can't answer definitively how it's going to be owned by quality or not. I guess, I mean, quality is you typically think of the offline measurements. So I don't know. I guess, Rob, maybe you're right that we need to have quality involvement if it's going to be in-process control. I just want to go back to a couple other questions that were asked before while I was muted, and because there was a question about how we choose our PAT, and to be honest, I would say because we started actively working on PAT and biologics only about 4 or 5 years ago here at Bristol Myers Squibb. So we've really gone after the low-hanging fruit, to be honest, because there is some low-hanging fruit in terms of the process has actually undergone a lot of evolutionary changes in the past 5 years. The titers are much higher. We've introduced elements of continuous, and really the analytics, in a sense, they really haven't kept up. Like especially with Tony was talking about continuous, asked me about continuous manufacturing before. We really need process analytical technology tools to make the continuous capabilities fully capable, because there are still some quite long steps during the elements of continuous where we're waiting for say QC test to come back to start the process. So those are the ones we targeted first. We're basically to decrease lead time especially for those semi-continuous processes, we can eliminate a quality offline method, and also process consistency, because so the titers are getting much higher, you're getting, and then the UFD, the final concentrations of our drug substance are getting much higher. That's introduced its own issues with process consistency, especially in terms of because as we've learned, the protein concentration of monoclonal antibody itself can affect excipient concentrations. It's something called the Donan effect. So that even just small, say your protein concentration is 210, say versus 200, that can significantly affect the concentrations of your excipients during processing, so that your end product is going to have significantly more variability. So all of a sudden, protein concentration becomes far more critical in terms of maintaining the consistency of that and its importance on just the overall process consistency and quality.
Jay, maybe to follow on that, do you, on the monitoring versus control question, it feels to me like the UFDF step should be prime for control, right?
Yeah.
I mean, there are a lot of, I mean, over the years, I've definitely heard companies talk about, you know, over-concentrated, under-concentrated. I think, Ralph, when you're talking about the final formulation, it's awful when, you know, it's the wrong concentration and you have to go back and figure out how you're going to reprocess a batch. So getting to control using like FlowVPX as a control for the UFDF step, do you see that, you know, how do you see that evolving at Bristol?
That's kind of where we're right now. I'm trying, when Ralph talked about value proposition, I'm trying to sell the value at present to our process team of process control for UFDF, and I think we have a pretty strong argument. And I should be careful when I tell this, but the process consistency, if you are not only monitoring by mass, you're talking about plus minus 10%. And as Ralph has shown, and we've demonstrated ourselves, you can decrease that probably to plus minus 3% by using inline A280. So that's a significant gain, and not just in terms of like protein concentration, like I said, but also the critical quality attributes of your excipient concentrations. And I also wanted to go back really quickly too, in terms of which instruments you'd like to combine PAT with the FlowVPX. And one suggestion was online LC, which I think Andres may have mentioned. And that is for a continuous process. Say you're measuring like a CQA like aggregation or your charge species. In order to have an accurate mass balance of your entire product pool, for example, you need to have an accurate concentration. So an LC system typically can do a separation in like 2 to 5 minutes to determine your CQAs. But of course, like your protein concentration is continuously changing. But you take snapshots of windows, you can basically come up with a model that can give you an aggregate of what the CQAs are based on the mass and their values. And that's kind of something we're working on actually, so that you can have a continuous system where you're monitoring your CQAs and what they would be, say, if you start and stop the collection at a certain point. So that's something that I think that an accurate mass balance is critical in terms of determining the CQAs for like a given continuous batch.
So maybe opening it up to everyone. But when you think about our industry and all the touch points we have, nothing is happens at just one site, right? And Rashmi, you see it obviously at KBI where you're developing processes. There's tech transfer happening coming into KBI, and just when you think about FlowVPX and other PAT technologies, how helpful will they be in your mind in terms of the tech transfer process? Because it's not simple, but I assume the more data you have and the more robust the methods are, the easier the tech transfer is going to be. So I'd love to hear people's thoughts on transferring processes into the KBIs of the world and the use of PAT technologies to help in that tech transfer process.
I'll just go real quick, or I don't know if I jumped the line, but I think it's absolutely critical as part of the value proposition that we sell this. And that's actually something I want to investigate when we're purchasing new instruments from Repligen and make sure that the quality control is very high, but that's one thing that we'll need to establish. And once we establish that, then we can have evidence that we can quickly transfer between sites, or not quickly, much more quickly tech transfer, say, a given process that relies on, say, a flow A280 meter. And it's like a complex, high concentration UFDF process. Much simpler to transfer between sites and locations. I believe it will be.
Yeah, so we haven't really used the flow VPE in the GMP environment. We have been using it in the PD labs, but as Jay said, if we have data to prove it, to back it, we can definitely transfer it. And I can also think of a couple of simple applications. Like for example, like we had a process where the flush for the viral filtration was dependent upon the concentration. We reach a particular concentration, we stop the flush. So things like that, I think I can see that easily being transferred if we have data that it is working well in the PD labs time and again.
Great, and maybe expanding on it a little bit. When you think about, there's been a lot said around, digital twins and digitalization of data. Maybe Andres, Ralph, do you want to comment a little bit on where you might see PAT technology falling into, the whole digitalization revolution that's going on and trying to create digital twins of processes?
Sure. So maybe one aspect that we think that is quite promising is to follow the efficiency of a column. So we know with digital twin how it behaves during time, and you can see, okay, now we're losing some binding capacity, and maybe it is time to either change the process parameters or maybe take a look if we need a new resin. So I think in that regard, the digital twin can be quite valuable for the process.
Very good.
Yeah, and I can add to that. Fully agree in the sense that a digital twin really, these concepts are getting more and more, say, routinely used, but very an important prerequisite for that is you need to have the data in order to build your digital twin, right? And I think online PAT tools such as FlowVPE are exactly there to provide the data so that that can flow into the models underlying into the digital twin. And I don't think that you can really establish digital twins by having offline sampling approaches and offline QC lab experiments. I think it's a very important part of that in order to establish digital twins.
Hey, Raph, I'd like to hear your thoughts on the last question about CMO transfer. I think you might have some perspective there.
Yeah, I think, if we talk really about tech transfer, right? So one important thing is that if you really move from one side to another one or from one skill to another one, sometimes there is really a lot of need for engineering runs or development work at scale to be done in order to fully understand how this product is behaving at specific site or a scale that you want to manufacture it at. So one approach would be to in an ideal world, we would all have the same equipment, right? So where we would transfer from one side to the other one, and there would be a 600-liter tank from the same brand that is put at the CMO, which is exactly the same. So we wouldn't need any development work to be done at all. But if in absence of that, online tools will really give us that understanding in the real time, where if we would otherwise have to do an engineering run to get data, it might be data we can get, let's say, directly as part of PPQ during a tech transfer, where, very concrete example, mixing studies, right? If you go from a 5-liter tank to a 50-liter tank, you would have to characterize how your mixing is going, but having your online protein concentration or any other PAT tool that you can track how your mixing is going, and it might not take 5 minutes how it would be on the 5-liter tank, but at least you can monitor and you could say, okay, maybe after 8 minutes, my mixing is completed. So it's this kind of information that otherwise would require a lot of development work that you can just get, let's say, in real time without any need for at scale tests to be done.
Yeah, maybe Ramsey, maybe back to you. Any questions from people online?
Actually, there is. I'm glad you reminded me. It says, has anyone presented data on the flow VPE or X to the authorities? And if so, what was the learnings? And I guess that means the health authorities like FDA or the EU.
Silence could be no.
Got to be Roth.
So we haven't shown, let's say, any data as of yet, but we're in the process of doing so. But we haven't done that so far.
Oh.
Yeah, and that's all I have from the line right now.
So maybe wrapping up, if you think, we can start with Rashmi on this. If you were to give advice to someone who's starting to implement Flow VPX, what would be your top 2 or 3 things that you would advise people?
Yeah, so, my personal experience about using this technology is that, it's a very, very powerful technology, but it took me some time to understand its full potential. So I would, if I had to advise, I would say that, understand the instrument, understand the technology very well. And once we do that, like, we can just extend the application to like no end.
Jay?
I would say be sort of like what along the lines of what Rashmi was saying. I would be very careful when you set up your methods and so like the data collection to be so that the data is useful, because it's a little it looks like a very simple tool like from the start, and you can just plug it in and start collecting data. That's actually one of the biggest issues we had early on in the days of PAT, was that people would just get a PAT tool and plug it in and not really go through like the proper analytical, so like the methodical like system suitability testing, so every week, every day, whenever, just checking the instrument is working perfectly, and you're using the most accurate method for that molecule. Because then what happens is you get data that doesn't look so good, or it's like you don't, the data hasn't been like timestamped correctly, and it's really difficult to go back and sort of make anything meaningful out of it. But it's just a very careful, especially if you're going to use it in a pilot plant or anything, to really carefully set the thing up and just so you don't generate bad data from the start, which can cause more headaches than anything else.
Great, thanks, Jay. Andres?
Yeah, maybe to add what has already been said, I would suggest to think about how you collect your data. Either you're using the electrical connection, or if you're using the OPC server, that you think beforehand how you can get the data, because the idea is to get that as fast as possible. And once you have your instrument, a lot of the times you have to wait until you develop something that enables you to get the data, and it can be a bit frustrating because you can see that you're getting your data, but then there's a delay before you can really use it to its full potential. So maybe invest beforehand a little bit in really thinking, okay, this is how I can access my data in real time, and how can I unlock this capacity?
And Raf?
Yeah, I agree with what everyone else said so far. Maybe one thing I can add is just that if somebody is about to implement FlowVPE or any PAT tool, I think tying that back to what we said in the beginning of this discussion was also it needs to be done by a dedicated team, right? Because there will be challenges to overcome, and there will be a lot of pushback from regulatory quality. I mean, people that in general are, let's say, afraid of implementing new things into a process of which they know that it runs very well, and why should we change that? So having really a dedicated team with people that have that mindset that they want to change something to even better, right? I think that's very crucial, because now looking back at the last couple of years that we have been implementing FlowVPE, if we would have had a team that would do this maybe on the side of their day-to-day job, I think it would have taken much longer, or maybe it wouldn't have worked at all, right? I mean, we really had people that were dedicating in a dedicated way working on implementing this, and I think that's one of the success, let's say, keys to success of having reached that implementation so far.
No, that's great. And what struck me when just listening to the presentations over the last month was, the applications are just so broad, right? I mean, we saw everything from DVCs to full finish applications. But I think the thing that struck me the most was the amount of data that you were collecting on every 10 seconds versus what you might have traditionally done, which is, send a sample off to QC, you wait 2 hours, or maybe you have to wait a day or two because of the complexity of the assay that has to be run. But doesn't it open your eyes then to the potential of not just FlowVPE, but any technology that can give you data in seconds? you think about the problems you can solve, stuff that you would never think about using it for. You now say, oh, I can get a result in 10 seconds. I don't have to wait for the quality control team to come back. It kind of broadens your horizon to the potential of the technology. And maybe that's my last question back. We maybe go through, start in reverse order with Raf. I mean, can you maybe speak to, was there anything that you saw that it said that you then decided, oh, we can do X that we hadn't even thought about, just in terms of the application of the technology?
No, you're absolutely right, Tony. I think there are two things that come into mind, right? I mean, one was what I presented last week was the filter adsorption. I think in order to get a data set like that, it would be nearly impossible to do with the traditional sampling approaches, or you would kind of have to do a very elaborate dedicated study and use a lot of material. So having that refresh rate or data at I mean 10 seconds, I mean with some optimization you can even bring it down to 5 seconds, right? So that's a lot of data that was never there, a lot of insight into the process. And before we actually plugged it into a process, we weren't even thinking that this is the data we were going to see. So I think that was really a very important learning. And that learning also translated into us moving from monitoring to control, right? Because before monitoring, we were saying, okay, let's put this online protein concentration tool in there, and we can replace the offline sampling. So we might shave off 2 hours waiting time for the QC lab analysis. But then having that, I mean, we could even just, now we have all of the data, why don't we use it really in real time to control the process? And that thinking only came from when we actually saw the data, we had it implemented into our process, and we didn't really think about that before we had it, let's say, available.
That's great. Andres?
Yeah, as Raf said, with this fast sensory information, you open the door to monitoring, so you can better develop your process, and for control as well. So you can actually control your process based on real information and taking into account the variability that's happening in your process, whether you want it or not. And maybe for our specific case, the combination with the moles was before the full VP. I mean, we were able to do it at the analytical concentration. So if we hadn't had access to this fast concentration information, this application would have remained in the analytical realm, and we wouldn't have been able to use it for a process stream.
Jay?
Yeah, that's, I mean, actually, I wanted to just mention it's an excellent point. So it opens up a whole other world when you get high quality data in seconds. The other side of the coin is this, what I call this fear of data. And I don't know how I want to open up, maybe Raf is smiling. And I've heard this, I mean, to me, it almost sounds counterintuitive, but it's actually not too, like, based on reality. That a lot of people are afraid of this data. They like, oh my God, they're gonna show, it's gonna show that our process is inconsistent. Which is, I think that's, people are a little afraid of that. And I'm like, well, that's FDA, they're gonna want this. We can't hide anymore. You can't just say, see no evil. So I think that's, to me, that's the argument I use, is that we can't hide anymore. We have to show them data. It's just as long as these tools produce like really high quality data. Like hopefully, I think the worst fear is people think, oh, because when you say do a QC assay, something doesn't work right, there's like these investigations. It's like, every time there's a bad piece of data, we have to investigate, and hopefully that's not the case. So that's what the other side is, overcoming like this fear that we're going to expose ourselves if we collect all this data and see that there's actually a lot more noise than we thought there was. Necessarily just being okay with that, because that's reality, right? It's like making sausage.
Great, thanks, Jay. And Rashmi?
Yeah, so before we implemented FlowVP for DBC determination, we used to get like a curve, a breakthrough curve with like 10, 15 points. But once we implemented FlowVP, we realized that we are getting this nice and smooth breakthrough curves. And, that opened up the potential application for, studying the resin lot-to-lot variability as well as resin lifetime study. So we have done this, we have used it for the resin lot-to-lot where, we are comparing a resin from the GMP and from the process development labs, and we just overlay the DBCs, and even minor differences can be caught. And we are definitely going to explore this for the resin lifetime study where we can see that the difference in the DBC curves, the breakthrough curves, is a very important tool to tell us how good our resin is performing.
And no one wants to run resin lifetime studies. It just takes too long. So, you know, I can see it being used as well to put in the cleaning the cleaning cycles on chromatography resins, and what the right time frame is to do that. Is it every 5 cycles, every 10 cycles? You can probably use this technology to help determine what the right timing is. Now, this has been great, guys. I just want to maybe close up. Any questions, Ramsey, from?
No, I think we can wrap up.
No, great. I just want to thank the 4 speakers. It's great that you all kind of shared your experience. Not every pharma company wants to talk about what's going on in their labs, in their departments, but honestly, the take-home message for me is the breadth of the applications. And just want to thank everybody for joining, spending the last 4 weeks, Ramsey's, for hosting this. I think you're probably going to make it an annual event at this stage, given the amount of interest in what's going on. I think the commitment from Repligen is that we're very much committed to PAT. Obviously, we think very, very highly of technology, hoping that this is the new wave of inline monitoring, at-line monitoring. And it's obviously much broader than just VPE technology, but this is probably the right start. Really, again, thanking everybody for joining us and sharing your experiences. I think that's incredibly valuable. And with that, I think we're good to wrap up, Ramsey. I'm not sure if you have any final comments.
No, just thanks a lot. Thanks everyone for all the hard work you've done over the years on this product. I know not always easy, and there's challenges, and and we're here to help too. So anyone who's on the line who's going down this road, reach out, and we're happy to help and get you through any tough times with the product.
Very good. Thanks, everybody.
Thank you.
Thank you.
Thank you.
Take care, everyone.
Yeah, thanks, guys.
Thanks for the invitation. Thanks, Tony.
Yeah, no problem.